Yeah, start again.
Hello, everybody. Those of you here in the room in Bozeman, and also those of you joining us online. Today we're going to learn a little bit about imaging microorganisms on surfaces, biofilms. I'm Phil Stewart, a professor in the Department of Chemical and Biological Engineering here at Montana State, longtime participant in the Center for Biofilm Engineering. I'm going to provide a little introduction and context, and then turn the show over to my colleague, Betsy Pitts, who's the Microscopy Facility Manager here at the Center for Biofilm Engineering. I wanted to tell you about our sponsors today for this webinar. They are the Montana Nanotechnology Facility, or MONT.
Look, you're up on the screen.
And we're going to stop.
Science Foundation supported sites around the country that make tools for fabrication and characterization of nanotechnological devices and products available to users not only on those campuses or sites, but also from elsewhere in industry and around the country. And the Center for Biofilm Engineering is also a National Science Foundation founded center, an engineering research center that is the oldest and best known center of its kind for biofilm science, education, and technology transfer. So today, the particular piece you're going to be hearing about is the expertise in imaging of microorganisms at surfaces that is housed physically in the Microscopy Facility at the Center for Biofilm Engineering. What I want you to get from the webinar today is an introduction to the physics and biology and practical tips for how to do this kind of imaging, and also the message that this facility, these resources are available to you, whether you're here on campus or an external potential user. The MONT umbrella under which the Microscopy Facility that CBE is part of allows us to offer these capabilities to, we hope, users locally and externally. So, what's a biofilm? Biofilm is microorganisms, think bacteria or yeast, that are banded together in dense aggregates, usually stuck to a wetted surface. The slippery slime on a rock in a stream is a biofilm. The deposit or encrustation that you can see on the inside of this urinary catheter is a biofilm. And the corrosion-inducing film on the inside of this oil-filled pipeline is also a biofilm. So one thing I want you to take from this slide is that we would say that biofilm formation is an ancient, ubiquitous microbial survival strategy. It happens in nature. We identify it increasingly in a number of medical contexts where they are the cause of persistent infections, and also widely in industry. The other thing I want to register here is the two essential constituents of any biofilm: microbial cells and extracellular polymeric substances that those cells themselves elaborate, typically polysaccharides or proteins, that form the hydrated gel-like glue or extracellular matrix that holds the biofilm together. And here's an example of that in a picture and a preview of the kinds of images that you'll be seeing more of in a minute here. On the left here in green are the bacterial cells themselves. This is a pseudomonad that contains a green fluorescent protein. And so the cell bodies are lit up green. They're just the actual cells themselves. On the right, in the same field of view, we're imaging in a different channel the results of staining with an amine reactive dye that's tagged with a red fluorophore. And there you can see this fabric-like web or mesh of material. This is the extracellular matrix, or the one component of the biofilm. And if we put those in overlay, you can appreciate that the cells, the green rods are actually mostly associated physically with that kind of scaffold, this extracellular matrix. Since we're thinking about nanotechnology and microfabricated devices, I want to just give a few examples of the sorts of systems that we might be interested in imaging. Here's one from a company called Sharklet that is manufacturing topographically modified material surfaces that they claim are anti-biofilm, that prevent or reduce the amount of biofilm accumulation. The structure of those surfaces is inspired by the structure of shark skin, and you can imagine that this would be a kind of material that we might want to examine for the presence, amount of microscopic surface. But certainly, if materials like this end up being effective at reducing biofilm accumulation, they could be very attractive. For example, in a medical device application, would help reduce the infection associated with the catheter, for example. Here's a technology from a materials science group at Stanford that has fabricated nanostructured molybdenum disulfide films. And these have the remarkable property of being able to take in a large swath of the visible spectrum of light, and in the presence of aerated water, generate reactive oxygen species. And they're using that, they showed they could use that to disinfect the water above this surface. Shine a light on it, and can disinfect the water. To a biofilm guy, this looks like a great potential anti-biofilm technology. If there's a bacterium on that surface, I shine a light on it, we'll be generating biocides, these reactive oxygen species, right at that interface, right at the attachment location, and have a good chance of being able to kill that cell and possibly, because of the oxidizing power, remove it. There's a lot of interest in using microfluidic devices in science and in designing new products. Here's an example of using one of these to study quorum sensing, the cell-to-cell communication that many bacteria use to orchestrate group behaviors. And what they've done here is to build a device with a primary flow channel that's along the top of this left panel here that has dead leg or side arms. And here you can kind of visualize the flow. They put some fluorescent beads in, and you see the streaks in the main flow channel there. You see that in the stagnant crevices come off of that main flow channel, there's much less fluid motion. And when this is coupled with a fluorescent reporter approach to imaging where the bacteria are actually doing this quorum sensing, which shows up in yellow here, very localized. It turns out that it happens in regions where there's still enough bacteria, but the flow isn't so strong that it just sweeps the signal away and dilutes it. So up in the flow channel, the concentration of the quorum sensing molecule never builds up to the point that it can trigger to a biological response. It happens partway down into these crevices that where the flow becomes a little bit quiescent. And here's an example from our own center of microfluidic droplets, really tiny, little tiny individual test tubes that have, in which a bacterium has been trapped. The bacteria are green, and these bacteria are able to, by their metabolic activity, facilitate the deposition, the precipitation of a mineral phase, calcium carbonate. So these radial structures that you see in some of the drops are in fact this mineral material. Here's one of those imaged under scanning electron microscopes. So the interaction of microbes with mineral particles is another place where kind of imaging we'll be talking about today could be of interest and application. as is the problem of microbially influenced corrosion. Here we're looking at the association of microbes with a metal surface. The image I'm showing you here is not fluorescence or light microscope image. It's a colorized scanning electron micrograph. But I wanted to show it because the facility that this was taken at, also right next door here at MSU in our imaging chemical analysis lab, is also one of the facilities that falls under the MONT umbrella. And this kind of microscopy is complementary to the fluorescence microscopy talking more about today. Here's a very quick overview of some of the capabilities at the Center for Biofilm Engineering. We have many reactor systems for growing and testing and interrogating biofilms. We have computational models for analyzing the behavior of these complex systems. And today we're in the middle here talking about the capability for imaging living cells at interfaces. And that's going to be the emphasis here for the rest of the presentation. I'm now going to turn things over to Betsy Pitts, who again is the Microscopy Facility Manager at the Center for Biofilm Engineering. Betsy individually trains our users and works with them to optimize and troubleshoot their research approach. She has many years of experience in imaging biofilms and microbial surfaces, and she'll share some of that with us now.
Thanks, Bill, for the introduction and for introducing biofilms and nanoscale projects. So that brings me up to talk about how we actually image biofilms, and perhaps it'll help you get a better idea of how you can combine it to what we do in some nanoscale projects. So how do we image biofilms? Most of our imaging is fully hydrated, so we can see what's happening without introducing drying artifacts. So we can use flow, so we can see activity. It's fluorescent because you want to see biofilms, see bacteria, not very visible otherwise. And it's collected with confocal microscopy because our samples are not generally thin and flat. When do you go to the confocal? Primarily, if your sample isn't thin and flat, and we'll talk about that later. When you have very specific fluorescence emission or excitation needs. If you want to use triple labeling, for example, that's a little tricky. Or if you want to generate an in-focus 3D look at your specimen. So let's back way up. How does a confocal work? And even further than that, how does a microscope work? Well, if you have a lens, the object has to be a certain distance away from the lens to be in focus, and that's called the focal plane. And you can't change it. It's a property of the lens. It's determined by the radius of curvature of the lens. So that distance is set and fixed. And so if you have a thick biofilm sample like we do here, the only thing you can do to see it is bring it to the focal plane. And then we can see it. And the depth of sample we can see in focus is called the depth of field. The problem, if you have a sample like this biofilm that's kind of thick, is that most of what you see is out of focus. And that's what we call out-of-focus-haze. And so if you're looking at this sample on a light microscope, most of what you see is out of focus. It's terrible. So you can either make your sample thin and flat, or like we do, leave your sample as is and change the way you image. So we go to the confocal. Excuse me. And here's why the confocal is so cool. We avoid all the out-of-focus haze. And this is a pretty simplified diagram of a confocal. We have a biofilm sample here. This one's been made to be green fluorescent. We have a simplified objective lens in the middle there. Got the focal plane, a beam splitter, which is pretty standard, and a detector pinhole. There are actually more pinholes in a confocal, and it's way more complicated, but we can simplify it. And then a light detector, photomultiplier tube in the back. So If I have a green fluorescent sample, I need blue excitation light. There it is. I excite green fluorescence. And now we have green fluorescence coming from all areas of the biofilm that's been illuminated. But the beauty of the confocal is that only the green fluorescence coming from the focal plane can get back to the pinhole. So that's all you see. It's that simple. It may be a little more complicated, but the principle is that simple. So all you see is what's coming from the focal plane. So you don't see a three-dimensional picture. You see a flat image. And here is an example of that. The image on the left was collected on an epifluorescent microscope. The image on the right is the same exact pair of fluorescent beads collected on a confocal. You see on the left here, you see out-of-focus-haze. You're used to interpreting that as topography, as three-dimensionality, but it's out of focus, and we can't see that bright, smaller bead very well. And the confocal image on the right is a cross-section of both beads, and it's beautiful. So what we do on the confocal is take many of those through our specimen. Usually it amounts to hundreds, every 0.2 microns perhaps, all the way through the sample. And then we put them together like this little movie shows to generate a maximum projection of our object. Or this kind of view. In this case, we have a maximum projection in the middle, and then what we call orthogonal sections. You can generate these orthogonal sections anywhere through the sample. In fact, the white horizontal line you can see in the background there, I put that kind of bisecting the big larger bead, so you can see a good kind of equator-sized cross-section of it. But with the vertical white line here, I kind of just shaved off a little bit of the big bead. So you see a smaller cross-section over there. So we can move those around and see inside our sample. Get really nice three-dimensional views. Or we can make a 3D reconstruction like this. Make a QuickTime movie. This is a three-dimensional object now, so we can rotate it in space, look at it from any angle. So with confocal microscopy, only the image of the focal plane is visible. We have to take optical sections through our sample and then put them together later in software to give us a three-dimensional view. You can tell right away this is a confocal image because the hallmark of a confocal image is that everything is in focus, but you also see depth and three-dimensionality. So you should be able to spot these right away. And here's an example of the kind of movie we can make from a confocal image of a biofilm. The bacteria are about a micron in size, so you get an idea of our typical resolution. Which is pretty good. So what parameters are most important for you to consider if you're going to do confocal microscopy of biofilm? And these are my top 5. There are others. Again, you can complicate everything, and it maybe is complicated in the end, but you can simplify it. Things like point spread function, there are aspects of confocal microscopy that are important, but not in the application day-to-day when you're trying to image. I love the fact that these 5 things are actually written on most objectives, so you don't have to wonder what the secret code is. Everything is there. The first one, magnification, is right here, and everybody probably knows that. This is a 60x objective. The next one is actually a quantity called numerical aperture, which is related to resolution. It's written on an objective. We'll talk more about that in a minute. The resolution in X and Y is always better than the resolution in Z. In fact, the resolution in Z is usually about 2 times bigger, which means worse. The immersion medium is written on an objective. This is a water immersion objective. You'll either see water oil or nothing, which means air. Cover slip thickness is written down here, and I have a green circle around it. There's a 0 on this one, which means you don't need a cover slip. And working distance. It's down at the bottom. It's always in millimeters. The working distance here is 2 millimeters. So a little more about resolution. There are many ways to calculate resolution. This is one of the ways that we use, a simple way. Resolution in microns or nanometers is equal to the wavelength of light used for the imaging divided by 2 times the numerical aperture. And we can calculate the numerical aperture. N sine of alpha. Alpha is the half angle of the lens. Gives us an idea of the light gathering capability of the objective. And N is the index of refraction of the imaging medium. If you remember physics, N is a ratio of the speed of light in a vacuum to the speed of light in a medium. It always slows down. So this number is always going to be bigger than 1, bigger than or equal to 1. And here at the bottom are the media that we can see through. So these are our options. So if we're trying to get the highest NA, the best resolution, we'd go for oil. So you might be thinking, great, I'm going to do all my imaging in oil, which is really a great approach, except we image our sample in the medium preferred by the sample. And in the case of biofilms, that's water. You always want to match the index of refraction above and below a cover slip if you're using one. So that will also tell you which medium to use. You get better, much better Z resolution when you do that. So we can see that our maximum index of refraction is 1.5. Our maximum NA is 1.5. And that gives us our maximum resolution for our kind of optical microscopy, which is about 0.2 microns. The working distance is the other quantity that I just want to clarify as critical, especially for biofilm imaging. Working distance is defined here at the bottom in blue. And pretty much if you put the lens, the working distance away from the top of the cover slip, whatever's right under the cover slip will be in perfect focus. So it's pretty much the same as the focal plane, but it includes the cover slip. But our favorite way to think of working distance is up here in red. It tells you how far into your sample you can see. It really does, because, for example, if we know this sample is 2 millimeters thick, just looking at the scale, if it's the scale, can you possibly see down here to the bottom of the biofilm? No, you can't do it. It's impossible. You'll never get there. Because to see the bottom of the biofilm, you have to raise the sample up to the focal plane, and you'll smash it into the objective and break something. So I think this is especially critical for us because our samples aren't thin and flat. So how far into them can we see? Well, you have to figure that out in the beginning. Is it important to you? Do you want to see down to the bottom? Is the biofilm so thick that you can't? Do you only want a top-down view? It really does depend. We don't always want to see all the way through and cross-section our sample. Here are 2 60x objectives that we have in our facility. What you do to determine which one is best is look at the working distance, which is circled in red on each. The one on the left has a working distance of 2 millimeters. The other one, the range of between 0.3 and 0.2 microns. So that one on the left, the one on the right, the working distance is too small. Wouldn't really work at all for this sample. And here's a table just to show you some of the standard objectives and resolutions in imaging media. The top 4 are pretty standard on light microscopes and confocals, and we have them all. And you'll see the trend we expect. Numerical aperture, and therefore resolution, increases with magnification, and working distance goes down, way down. And that just means what we already know. If you want to get better resolution, you get closer and you collect more light. And also, we know that an imaging medium encourages the light to bend towards the objective. We collect more light. So imaging medium gives us better resolution. And then down here are the objectives we actually tend to use most often to image biofilms. And they're all, you'll see, long working distance water objectives. And they're really different. The resolution drops a little bit, but it's good enough to see bacteria. So if you're thinking about a nanoscale project, these things are pretty important to, look at nanobeads. The confocals we have at the CBE, we have two Leica TCS SP5 confocals. We have an upright and an inverted. And the situation I have shown here, we put our biofilm covered surface in a small petri dish. That's one of our standard ways of imaging. And with a long working distance water objective on the upright, we come in and hold a 2 millimeter column of water and image that way. We can see, at maximum 2.2 millimeters down into the biofilm. On the inverted, we use cover slips with a glass, we use dishes with a glass cover slip bottom, and we look through the bottom. The working distances on those objectives are very small. You can imagine it's pretty hard to hold a 2 millimeter column of water in the inverted situation. We don't do that. And you might say, well, why do I care? How do I know which confocal to choose if I can see all the way through the biofilm? And that's a really good question. You can't. And that is a huge consideration when you're imaging thick objects. This is a confocal time-lapse movie of a biofilm in a flow cell. And what we're doing here is changing the osmolarity of the media as it flows over the biofilm. So we're seeing it shrink and swell with salt concentration and then without it. And this looks fantastic, doesn't it? What a wonderful movie of a biofilm. It is cool. But you can also look at it from the side here. This is the exact same confocal stack viewed from the side, and you see that that large cluster appears to be hollow. We know it isn't. We've shown this in many other ways, like with cryosectioning. They're generally solid. This just means the light is attenuated maybe 25 microns into the biofilm, and we can't see anything anymore. So this is what we call a shadowing artifact. You can see the biofilm shrink and swell, but this is a good example of shadowing artifact. But the movie I showed you earlier was fine. You wouldn't know there was a shadowing artifact. You can see really nicely from the top. The laser lines we have available, we have 4 lasers. They're pretty standard on confocals. 405, an argon ion with 4 laser lines, a 561 nanometer laser, and a 633. And you see here I drew red circles up there. In this case, I turned only 3 lasers on. This is a screenshot of the control panel on a confocal. With these lasers, those are pretty standard. Most dye and stain manufacturers actually produce dyes and stains with those lasers in mind, like the Alexaflor series, Alexaflor 88, Alexaflor 633. So we're good on lasers. You just have to find the right fluorescent probe for your sample. These are some of our favorites, not a complete list at all, but some of the dyes that we routinely use and what they bind to, what they show us. Activity, matrix components, total cells. And here are some examples of images we've taken on our confocals using multiple fluorophores. The one on the left, we've used red and green dyes to show us, give us an idea of what bacteria in this biofilm are live versus dead. In the middle, we're using the absence of stain to tell us something. So if you watch this movie, we're watching Listerine work its way into a oral biofilm. And we see the loss of fluorescent color, which is a really interesting way to use fluorescence. Let me play that again. And then on the right is an example of biofilm on fabric, kind of interesting surface. Using multiple fluorophores at once can be really tricky. Not only do they need to be specific in what they're binding to or the information they're telling you, but actually separating the emissions on the confocal can be a real trick. So back to that screenshot of the control panel, if, for example, you were looking at 3 fluorophores at the same time, 3 lasers are on, here's why it can get tricky. I've drawn a yellow circle around the 405 laser. Let's say we're using that to excite a blue dye. Look at that blue dye, it's really commonly used, DAPI. The emission spectrum is a whale. It's huge. We talk about it as being a blue dye, it's also got green in it, it's got a red tail. So if you use DAPI with another fluorophore, you set yourself up for some challenges right away. We have ways around this, but it's tricky. We call these our detector slits. We can adjust the emission bandwidth we want to look at. But if you look at that area around 50 in the middle, where would you put that bandwidth so that you can get a clean picture of the green emission? It's not clear. You have to do you have to use some tricks to do that. We have them though. We have complete flexibility in the bandwidth we can look at, number one, that's huge. We can do sequential scanning. On a confocal, the imaging is also cool because you can use 3 lasers simultaneously and collect the emissions separately and simultaneously. You can also do sequential scanning, which means instead of using all 3 lasers at the same time, you can fire them really fast, one after the other, and pick off the separate emissions. And that can usually work around an emission spectrum like DAPI. So we can work around crosstalk and read through that way. The other one that I really like is spectral separation. And spectral separation is something that comes on board the software. And it's been used in what's called classy FISH. So classy stands for combinatorial labeling and spectral imaging. And FISH is fluorescence in situ hybridization. But basically, so you might ask the question, how many fluorophores can you look at the same time? 3? As many detectors as you have? 6? 5? Well, if you use 5 fluorophores and combine them, you should be able to come up with an infinite number of combinations that you can see. And that's what classy FISH is about. If you can separate the spectra of the dyes, you can do that. So what I have here is an example. I've done it on our confocals. We haven't applied it to the, we're going to a wonderful experimental example yet, but I have beads. Each one of these beads shown here has a green dye on its shell, and then another green dye with a very similar emission spectrum inside. So to our eyes, they're both green. We can't separate them. They use the same excitation, and their emissions are very close. Here are the 2 spectra. So what we do is highlight an area of a bead that we know is one contains 1 fluorophore, collect its spectrum, and do it for the other area. And then we take a picture of the whole sample and tell the software to separate the spectra. That's pretty easy. Here's what we get. I've turned one of the green colors red, just so you can see the difference. They're completely separated. And you couldn't get any closer than that. 2 green dyes with very similar spectra. Just to zoom in, here they are, separated. It looks like there's 1 bead that's all green, but ignore that one. So spectral separation is very powerful, and we've used it, a couple of us, with bacteria, with 3 different cytodyes together, 3 green cytodyes. It works successfully, very easily. So multiple fluorophores is definitely something we can do easily. So I'd like to finish with some applications, examples of how we use these techniques on real samples. And the first one is reflection imaging. So all the confocal imaging we've talked about till now has been about fluorescence. And that's the bulk of the imaging we do. But there's another way you can use the confocal, and that's with reflection imaging. You can hit the sample with a 488 laser, and this works well because that laser scatters most efficiently, and collect the reflected laser light and generate an image of the sample surface. Works very well on things that reflect well, not so good on glass beads. The light gets in and bounces around and makes kind of a mess, and not very good on biofilm, but the surface the biofilm is on is really nice. And here's an image that will help you understand the importance of reflection. This is an image that my colleague Lindsay Lorenz took of biofilm on fabric, and you really can't see the fabric. And so the image is not that useful. We want to see the fabric, but it depends. Is it polyester? Is it nylon? Is it cotton? We can stain it, but in most cases that would overwhelm the signal from the bacteria. So we're often stuck with this. I want to see the background, but I can't let it overwhelm the rest of the information. So she used reflection on this sample, and there's the complete image. She turned the reflection signal purple just to give us some nice contrast, but that's a beautiful example of how important the surface is too. And here's my favorite example. My colleague Heidi Smith used this technique in her PhD work to demonstrate a relationship between Antarctic sediment and attached microorganisms. So these look like SEMs, but they're not. They're confocal reflection images of sediment surfaces. And without the surface, without an image of the surface, it would have been impossible to get the data that Heidi got, or to be so convinced that these microorganisms are attached. So reflection is a beautiful way to augment fluorescent information with biofilms. Another application, curved surfaces. Curved surfaces can be really off-putting because they're not thin and flat, but we know how to image them. Here's an example, a catheter on the right, still in its sterile wrapper, and then Jennifer Summers currently has a reactor system going. You can make the catheter out barely in the falcon tube there. She's growing biofilm in there. And one of the issues when you look at curved surfaces is we're looking usually at the inside. Not always, the outside is fine too. When we do that, here's how we image. We generally cut tubing in half, then we have to image it this way. Take optical sections down one side, across the bottom, and up the other side. So hundreds of images, then put them all together. And Steve Fisher, who's done that, here is what you would see if you took this collection of images. Stripes that mean nothing at the time, but when they're put together later, you get this kind of image. When tubing is microbore, then you'll actually see a U-shaped cross-section. This is pretty curved, but this is a small section of it, so we can zoom in and see the biofilm too. The third topic that I think deserves attention is how you prepare samples for this kind of imaging. We often use small petri dishes, and you may be able to see on the right, I've got a piece of double-sided tape on the bottom of that petri dish. We actually have to stick our samples down or they'll float. This tubing has a small inside diameter, maybe 2 millimeters. It's dental unit waterline, so we've cut it in half, stuck it down, and then we submerge it in water and use our long working distance objectives. On the inverted, we use these petri dishes and look through the bottom. We often look at samples that are not stationary when they're submerged in water. like algal biofilms, or we've imaged bacteria attached to plant roots. And the struggle there is that the samples are waving the whole time we're trying to image them. And what we'll end up with is what looks like a strobe image, because we've caught the sample moving at every optical section, and it's impossible. You can't see anything. The image is terrible. I don't have too many because we usually don't keep them because they're terrible. But what we do to avoid that is put samples in small dishes like this and pour warm, clear agar over them. So embed them in agar gel. We can see all the way through the gel, and our samples are stationary. We see that that doesn't seem to introduce any artifact. The samples aren't flattened out. They still have all the topography. And then the last application is time-lapse. We are set up on both confocals for time-lapse imaging, and that to me means we have environmental controls. We have made stage modifications to hold flow cells. Immersion medium is an issue. If we're going to use the upright confocal in a dish of water, we'll have enough water to let us do a long time-lapse image. But if you're using the inverted confocal and you have a drop of water, it's going to evaporate. So we have a medium that is has the same index of refraction as water, which doesn't evaporate as quickly. We can do many different kinds of scanning over time, tiling. We can take Z-stacks over time, which is a lot. The images are huge, but we can do it. We can also do mark and find. That is, mark maybe 6, 7, 8, 9 different spots in an image, come back every hour and take a different stack at each one of those spots. Here are pictures of the incubators we have on our scopes. The left shows the inverted microscope. With a small environmental chamber, we can control the CO2, the oxygen, the humidity, temperature. That system is designed mostly for static imaging, small samples. Although we figured out we can take the bottom out and add plumbing, so small flow-through systems will work. And then on the right, we have the incubator on the upright confocal. That's a giant box that fits over the whole microscope. And it's difficult to do, but we've gotten the temperature up to 50 C in there for time-lapse imaging. That's more designed for flow-through systems. And here are some examples, some movies. First, neutrophils engulfing Staph aureus bacteria. So these are the guys in your immune system that protect you from invasion. Another one, a Staph aureus biofilm expressing green fluorescent protein in a flow cell during treatment. So being able to watch this happen is pretty powerful. We can often go in later and scrape a surface and do plate counts, but watching it happen on the confocal is pretty powerful. Here's one more. Time-lapse imaging is quite a big component of what we do at our confocals. To finish this off, we do a lot of image analysis. We can, I think, measure anything that you can photograph. If you can observe it and find the right probes to highlight the aspect of interest in your biofilm, we can photograph it and we can measure it. Here's an example. This citation, Kristen Brillier, for her PhD work here, she's now our lab manager, used our particle tracking software to show taxes toward hydrogen gas That was a pretty computationally intensive effort, but we have the software to do it. We routinely measure anything else you can think of: volume, area, red versus green. So I would say confocal microscopy is qualitative and quantitative. You can see it, we can measure it. Gene expression, fluorescence intensity, growth, accumulation, removal, different components, matrix components. And yeah, what we learn is, I think, from our calculations, from our quantification, and also just from watching movies, from looking, from seeing three-dimensional objects. I hope I've given you some idea of how you can use what we know in combination with some nano-inspired projects. And if you would like more information about our facility, here is my contact information. Phil and I would also like to encourage you to complete a survey on this webinar. Here's the link.
And I'll just come up here to join Betsy in thanking you all for your attention. We're going to invite your questions here momentarily. I just want to reinforce what Betsy just said, that this capability in the microscopy at the Center for Biofilm Engineering is available to you in partnership with MONT, and Betsy is the best place to make that inquiry about how you can access that capability. And we would genuinely appreciate your taking time to fill out the survey. So paper copies for the folks here in the room, online. Follow the link if you would. And it's short, and it will help us in fulfilling our reporting obligations to the National Science Foundation. You would complete the survey for us. We have a few minutes for questions. Good job.
Hey, thank you, Betsy. Excellent talk. I'm interested in inverted confocal. Do you have an immersion medium there? I mean, do you use?
With the inverted confocal?
Yeah.
You mean like if you have a dish with a cover
Do you
slip
put some
bottom?
water droplet on top of the objective lens and kind of?
Yes, absolutely. We have glycerin, oil, and water.
In the inverter, so
Yes,
so
correct.
they don't go right into the.
No.
The gravity always works that way.
Yes, but we use such a tiny amount, which is, everything is kind of a balancing
Yeah,
act.
that's the point I was trying to
Yeah.
come. So you put such a tiny amount, so you don't have too much time to go Z direction. To,
You do, Risha, you have quite a while. It depends on the medium. With oil or glycerin,
you go
easily
like
an
a
hour.
millimeter or 2 millimeter.
Well, that's the resolution is much lower on those objectives. It's about, they're usually on the order of 130 microns. So you can
I see.
only see 130 microns in. It doesn't include the cover slip,
I mean,
but
you can do slices.
Absolutely.
Within, I don't know, a couple micron steps all the way to.
it depends on, but the gel I showed you, those gels are usually 400 microns thick. I can see all the way through them,
Cool.
but I use like a 20x. I go down in magnification, but we do have the whole range. I think we have one that would suit you.
Well, yeah, you actually done something for me. I'm glad. I mean, it was wonderful.
Okay,
So we
good, yeah,
remember the
let's talk.
fibers with the
Oh, yes,
E. coli.
the glass
It was
fibers.
very similar to what you
Yes.
showed there.
Yes,
Thank you.
yes, you're welcome. Dave?
So a related question, if we were providing our own substrate and we wanted to match the thickness of the cover slip to your objective lenses, are there some standard thicknesses that are going to be appropriate?
That's a great question, yes. Also written on our objectives, it's a cover slip, when there is a cover slip thickness, it's 170 microns.
Okay.
So all of our objectives are corrected for that thickness. So yes, that's the one you want to use.
But again, one of our favored ways to do this is to get rid of the cover slip entirely and come down from the top into water with a water immersion objective, and then there's just an objective water phase in your material,
Yeah.
and that's all there is. And that's as clean as we can get this. Very compatible, having live cells in there as well.
I was inspired by your sharklet surface and thinking if you wanted to see right at that surface, it might be easier to come up from below and see the colonization or the disassociation at the surface. And of course, it would depend on if you can see through that structured layer and how well index matched that was and
Right.
scattering and all that.
Yeah.
But
Oh, we'd love to try to, you know, I'm proud of the fact that rather than modify our samples, we've really focused on modifying our facility to include everything we can find that will help us see your sample, whatever it is. And I think we have an objective situation. Sometimes we get both confocals going, go back and forth, see which one works best. So, yes.
Betsy's seen lots of people come in with requests, you know, hey, can you image this? And instead of just saying no, she'll say, I'm not sure, but we'll try. And so we're happy to do that.
We have a lot of fun and we learn a lot, and all of our wonderful colleagues, can I go to the next slide, Phil? Really, I think generate lots of creative ideas that help us keep expanding our applications. So,
Do we have any questions at a distance? If you do, you can unmute yourself, or if you do not have a microphone, you can chat your question and we can ask them that way.
So in addition to cell growth, can you also see nucleation growth of secondary minerals and begin to identify other solid phases that might be in your flow chambers?
Yeah, so the reflection mode has allowed us to visualize some of these mineral crystals. I think you've also gotten fluorescence out of calcite. Is that right?
we have it, and you can say, I've got it, but actually, there's some of the our graduate students and postdocs that I've worked with have done that. But yes, we have. It's a little tricky. Sometimes they're fluorescent. It depends on the mineral. Sometimes we can use that. And reflection can be tricky with crystals because, you know, light goes in and bounces around and comes back out, and it's kind of messy. But we've been fairly successful. Yeah, I would say yes. It's worth a try.
So I do have one question. Does sample bleaching become an issue when using time lapse? If so, how is it corrected for?
Well, most of our time lapse. Can I just talk to answer?
Yep.
Oh, most of our time lapse, we don't do with staining because in our experience, most fluorescent stains and dyes are toxic. So we use fluorescent proteins and do our imaging in time lapse with cells that express fluorescent proteins, and we don't see significant bleaching. My experience has gone up to 72 hours of time lapse imaging. I also do controls where I image with no treatment and just watch what happens and measure, quantitatively measure the bleaching. And it's insignificant enough so as not to impact our results. So I guess that's how we avoid it. We do not use dyes or stains. We use fluorescent proteins.
Anything else?
And not that's coming through.
Well,
One more.
there's one more here.
So one more question for users who want to come in and take advantage of the imaging capabilities. I'm assuming that the instructions on how to get started are online. Is there also some kind of billing information? Or, I mean, if I want to do a time-lapse study that's going to occupy the machine for a long time, are those sorts of parameters clear on the web someplace where we can get our parameters, what it would cost?
We can't really provide a formula quite. What we, if you're interested in talking through what you're trying to do with Betsy is the right way to go. If you're here on campus, we can train a student or a postdoc or somebody and charge, there's a certain charge for the way we do that. If you're in a company and an industrial project, we run a lot of those as well. But those, it's going to be a different model for how that gets put together. We really have to talk about the overall objective of it before we can start to.
So send an email and start a conversation.
Yes.
Send an email and start a conversation. And Yeah, so we, I guess we take it from there, yeah.
Okay.
I don't need to say more. We train everyone, I train everyone individually. We usually actually start with a proof of principle experience, where we see if it can be done, and then we go from there. That's the best place to begin.
Cool. Okay, well, thank you all.
Yes,
Thank
thanks
you.
for coming, you guys.
I'll collect these surveys and thank you very much for